So it's a great pleasure and honor, in fact, to introduce the keynote speaker of this session, Dr. Drew Pardoll, who is an international leader of immunology and immuno-oncology. So Dr. Pardoll is a professor of oncology, medicine, pathology and molecular biology, and genetics at Johns Hopkins. He's also the director of the cancer immunology program at the Kimmel Comprehensive Cancer Center at Hopkins, and he's the recent director of the Bloomberg-Kimmel Institute for Cancer Immunotherapy that was founded this year, or maybe last year, through a joint venture between former Mayor Bloomberg and Sidney Kimmel. So Drew did his MD PhD at Hopkins in 1982. He then stayed and did his medical residency and oncology fellowship at Hopkins, which he completed in 1985. He then worked for 3 years at the National Institutes of Health as a medical staff fellow, and then was recruited back to Hopkins in 1988, and then he has been on the faculty since then. In 1998, he became a professor of immunology and oncology and started to lead a phenomenal cancer immunology program. And so Drew made enormous contribution to immunology, many of which, in fact, have shaped immunology textbook. Among them, he discovered this population of gamma delta T-cell, which is a population of innate lymphocytes, which play a key role in antimicrobial immunity, and we learned recently probably play also a key role in anti-tumor immunity. He also discovered another population of innate lymphocyte, NK T cells. He worked for many years on understanding the regulation of dendritic cells, a population that, in fact, we study a lot in my lab, that regulate an immune response and contribute to induction of anti-tumor immunity. Drew has invented several cancer vaccines, including the GVAX and the listeria-based cancer vaccine that had led to significant clinical responses, in fact, before the checkpoint blockade. He is the inventor of something like 40 patents, most of them focused on immunotherapy. He discovered one of the two ligands of PD-1, of the PD-1 receptor, and had led the Hopkins Cancer Immunology program that developed the anti-PD-1 antibody, demonstrating their clinical activity in multiple cancer types. He serves on several scientific advisory boards, including the Cancer Research Institute, UPenn Gene Therapy Institute, the NCI, the Dana-Farber, the American Association of Clinical Oncology, and the Tisch Cancer Institute at Sinai. So he wrote more than 300 articles and reviews that contributed to shaping immunology and contributed to the current understanding of immunology, but also contribute, I think, to making immuno-oncology as part of cancer patient treatment. So he is he's also a great colleague, very attentive to trainees, but also has a clarity of thought and an independent thinking that has been, I think, critical for the field. So recently, I was attending an international cancer immunology meeting, and Drew was satirizing a presidential nominee, explaining how he himself can fix cancer and make patients great again. And we were laughing in the room, and there were two fellows sitting next to me, and one of them was telling the other, I'm not sure why they laugh. This is Drew Pardoll. I mean, I think he can do it. So this speaks, I think, to Drew's stature or to the credulity of the voters. Drew, I'll let you choose. So please help me in welcoming Drew Pardoll to the podium today. Thank you, Drew.
So first, I want to let everybody know that Miriam is not only admired and beloved here at Mount Sinai, but really throughout our whole field. She is, so it's really an honor to be introduced, even in an embarrassing way, by Miriam. I should also say, with regard to all of those titles, one of the things that I've learned is that the more titles you accumulate, the less scientifically productive you become. So I'm actually trying to get rid of some of those titles. But so I spent a lot of my time asking this question, which is how much cancer can we cure with the immune system? If you were to ask 100 oncologists 8 years ago, that question, the answer from 99 of them would have probably been between 0 and 1%. And now that's certainly changed. The door has really been swung open. And I think this, finally, the advances that have been made in cancer immunotherapy really validate a notion that immunologists have had for a long time, which that in many ways the immune system should be the perfect anti-cancer weapon. T cells and B cells, the elements of the adaptive immune response, have this unique system for creating incredibly diverse receptors. Roughly 10 to the 18th to 10 to the 22nd different receptors, which is actually more than you have lymphocytes in your body. So that diversity offers the opportunity for extreme specificity. T cells and antibodies can distinguish the smallest organic group, the methyl group, between two molecules with affinity differences up to 3 logs. And then the weaponry of the diverse cells in the immune system is really quite powerful and itself diverse, which makes it difficult to impossible for any given cancer cell to develop resistance to all of these cytocidal mechanisms that the immune system has. It took a long time to convert that concept and what we learned into successful cancer therapies. And things don't happen overnight, but I think there are always milestones in a field that kind of mark a change in paradigm. And I think for us in the field, it came at the end of 2013 when Science, in their last issue of the year, named cancer immunotherapy as Breakthrough of the Year. And actually, I made a comment to a phone call with a reporter for the Wall Street Journal, and this appeared in the Wall Street Journal in January. This is the only high-profile journal that I've ever been able to publish in without being brutalized by the reviewers. So I actually like that journal. I think that, I think this is, I actually worried when this actually made it into print that it was going to be hyperbole and overstatement, but I think it actually provides a mandate for our field that we really should work to try to achieve. Breakthroughs in therapeutics are never really breakthroughs. The clinical successes that have been coming over the last few years, really at breathtaking pace in cancer immunotherapy, appear like a breakthrough. But when you really go back and look at the origins and the basis for these clinical advances, you realize that the clinical breakthroughs have been built on a lot of science that came before it. So I put together these timelines of basic immunology and cancer immunology advances. Here in the top are clinical immunotherapy milestones. So the first thing that you see is that the history of cancer immunotherapy, the notion that you could get a patient's own immune system to attack their cancer, is actually very old. Coley's toxins are more than a century old, developed by William Coley here in New York. But the concept, which was absolutely correct, was ahead of its time and ahead of the science. So there was really no significant advances that occurred for 60 or 70 years. But then modern immunology began to really dissect how the immune system recognized antigens and how it was regulated. So you had monoclonal antibodies in the mid-1970s. The elucidation of tumor-associated antigens and what they were, this actually extends because we now understand that mutations can generate tumor-specific neoantigens. the elucidation of how T-cells recognize antigens, and then finally the elucidation of immune checkpoints as these critical inhibitors of down modulators of immune responses. And really, these had to be in place before we started seeing clinical benefits. First, the demonstration that systemic IL-2, not the easiest treatment to give or to receive, but could induce remissions in melanoma. Steve Rosenberg's proof of principle demonstration that expanded and ex vivo activated tumor infiltrating lymphocytes given back to patients could induce remissions. But then came anti-CTLA-4, anti-PD-1, having had the chance to be involved with the group at Hopkins that developed this, and in particular, Suzanne Topalian, who led all of the clinical development, seeing responses now not only in melanoma, but in kidney cancer and lung cancer, and now in 10 additional cancer types, the advent of chimeric antigen receptor transduced T-cells. All of this happening in medical terms, really a heartbeat, was built on the shoulders of really immune science. So I think it's congratulations to Mount Sinai's vision in developing an immunology institute, because really across all areas of medicine, the immune system is really central. I actually tell the medical students that there are very few things that bring a patient into the hospital or to medical attention that don't in one way or another involve a component of the immune system. So until about 10 or 15 years ago, I think immunologists viewed immune responses in a very linear fashion. Basically, immunization was an accelerator. There was signal one, which was the antigen, and then there was a whole host of co-stimulatory signals that led to an immune response. And what we began to learn in the 2000s is that, in fact, these accelerators are balanced by inhibitory signals, the brakes. They're often now called immune checkpoints. CTLA-4 was one of the first, others to follow. And there are also cell types like regulatory T-cells in which the whole cell is devoted to down modulating and regulating immune responses, subsets of myeloid cells as well. So in the 80s, 90s, and early 2000s, the focus for cancer immunotherapy was really pushing on the accelerator, essentially developing vaccines. And for various reasons, those vaccines, when used as single agents in patients with advanced cancer, were not successful. And the real breakthroughs came from approaches, therapeutic approaches, that inhibited the brakes. This actually was quite amazing to me, the notion that you could actually inhibit one immune checkpoint and get dramatic regressions of tumors in not insignificant numbers of patients, as we've seen with blockers of the PD-1 pathway. That told us something very important that I think neither the immunology community nor the oncology community, frankly, nor myself, really totally believed, certainly questioned, which is the notion that patients with cancer, large numbers of patients, large proportions of patients with cancer, really do have significant repertoires of T-cells in their body that can recognize their tumor and kill their tumor, save for a few of these checkpoints. To me, that clinical finding really opened up this amazing insight into the relationship between a patient's immune system and their cancer. So there are many, many of these ligands and receptors that either co-stimulate or co-inhibit immune responses. They're called co-inhibitory or co-stimulatory because they don't really do anything until there is recognition of antigen by the antigen-specific receptor. I'm going to focus on T-cells here. That's the only part of the immune system I really know anything about. But Miriam's trying to teach me about these myeloid cells. And that's signal one. And signal one then is modified by all of these other signals. If you knock out the genes encoding these various molecules and look in depth at immune responses in those animals, their immunologic phenotype is all different. These are not redundant. They're partially redundant, but the closer you look at their immune responses, the more you realize that they each play their own roles, some in combination with each other. I don't know how the immune system keeps it all straight, but these are turning out to be very, very exciting targets for cancer immunotherapeutic agents. The two that have now made it to FDA approval are antibodies against the first major checkpoint to be identified, CTLA-4. That really came from pioneering mouse studies from Jim Allison and colleagues. And then the second is PD-1. And certainly the PD-1 blocking antibodies, of which there are now, I think at last count, 14 in the clinic, are really driving a lot of the field. But this is really just scratching the surface. As I mentioned, these checkpoints play different roles in the immune response. So if you just take CTLA-4 and PD-1, the two that have been targeted successfully to FDA approval, they work at very different points in the immune response. CTLA-4 down-modulates early stages of T-cell activation, so-called priming. If a T-cell is ultimately primed, namely the co-stimulatory signals essentially outstrip the co-inhibitory signals, those T-cells will proliferate, they'll become activated, and if they're specific for a tumor antigen, they will traffic into the tumor. And in a lot of cases, you find that tumors overexpress the ligands for PD-1. In solid tumors, PD-L1 is probably the major ligand, although PD-L2 looks like it may play a somewhat unique role in certain tumor types as well. And the activated T cells have turned on PD-1, the inhibitory receptor, and these interactions within the tumor microenvironment inhibit these activated cells from ultimately killing the tumor. And that's how, in cases where you see a lot of PD-L1 expression in tumors, that these tumors have essentially evaded or resisted being killed by T cells that are in the body, that are in the tumor itself, able to recognize the tumor, but yet they don't kill it. This is a very dynamic process. So PD-L1 is not expressed constitutively in most tumors that are quote-unquote PD-L1 positive. It's actually expressed in an adaptive fashion when the tumor senses that it is under threat by lymphocytes. How does it do this? Activated lymphocytes that are a threat to a tumor, be they Th1 CD4 cells or CD8 cells, make gamma interferon. Gamma interferon actually will induce PD-L1 upregulation on the tumor. This is essentially co-opting a physiologic negative feedback inhibition mechanism that we use so that our immune responses don't cause too much collateral damage, that the tumor is now co-opted to protect itself. So here in this melanoma, you see CD8 T cells coming in from the top, and very few CD8 cells here. If we were to look at this biopsy a week later, or maybe even a few hours later, these CD8 T cells might have gone further into the melanoma. But if you now stain for PD-L1, you can see that it's largely upregulated on the surface of cells in a patchy fashion in the tumor, exactly where the CD8 cells are. So we call this adaptive resistance or adaptive expression. And if you actually look by messenger RNA, you find that this is where there is gamma interferon being produced by the T cells, whereas here there aren't any activated T cells, so there's low gamma interferon and there's no surface PD-L1. So how does this look clinically? The most amazing, to I think the oncology world, story that has come out of cancer immunotherapy is not so much the frequency of responses, although in some cancers it's very, very high, but it's actually the durability of the response is what you get once you get a response. And this was first seen with anti-CTLA-4 in melanoma, and there's about an 18 to 20% of patients that are now living to 10 years out from their initial treatment. These are patients with metastatic melanoma at the time of treatment. Prior to, if you go back to the textbooks in the 1980s, 1990s, early 2000s, they'll tell you that the 10-year survival for metastatic melanoma is about 3%. In second-line therapy, anti-PD-1 looks like it's plateauing out at about 35%. And these are studies led by, from the original studies, led by Suzanne Topalian, and now not as far out, but the first studies led by Caroline Robert of anti-PD-1 used first line in melanoma. So it's the first treatment that a patient with metastatic melanoma receives. Looks like it may be plateauing out at around 50%, which is unheard of, absolutely unheard of. Now we'll see where this ends up, but it's, again, the patients that don't respond don't do a lot better than with chemotherapy. They do a little bit better, but it's the responding patients that respond durably, and this, we believe, is immune memory at work. The notion being that our lymphocytes in our body are not just one drug, but they're literally thousands of different drugs that can keep on top of the tumor. And so when the tumor genetically evolves to evade the immune response, the immune system keeps up. Nonetheless, we can't forget that even if this plateaus at 50%, that still means that at 5 years, half of melanoma patients will have died. So we certainly are not ready to declare victory. This is what you see in lung cancer. Again, at one level, the 2-year survival rate of 24% compares with a 2-year survival rate of about 5 to 8% for chemotherapy and even targeted therapy. But that still means, and it probably looks like it's plateauing out at around 15%. That plateau for metastatic lung cancer prior to immunotherapy was more in the 2 to 4% range. But still, again, at 4 years, 85% of patients will have died. So I still think that, so this is why I spend a lot of time asking, how much more cancer can we, how much more of the immune response can we mine to treat cancer? So there's a lot, because there are many, many molecules that are still being discovered and analyzed that regulate the quality and amplitude of immune responses. And many of these are selectively expressed in the tumor microenvironment. This is just showing a bunch of them and also making the point that there are many cells in the tumor microenvironment, not only the cytotoxic T cell that everybody thinks about, but there are helper T cells, Treg cells, dendritic cells, macrophages, myeloid-derived suppressor cells, or immature myeloid cells. When you start looking at different tumors, different of these molecules and different of these pathways are overexpressed and others are not. And and there's a lot of diversity, variability from tumor to tumor. And so this begins to introduce the notion of looking in the tumor, looking at biopsies in of the tumor for biomarkers to help guide therapy to choose from a potential suite of drugs. This is our version of personalized cancer immunotherapy. And in fact, the first proof of principle is in fact with PD-L1 itself. So this is a number of different studies looking at the response rate, various different cancer types, and the key here is to look at the response rate in patients whose tumors by immunohistochemistry are PD-L1 positive, which means greater than depending on the test, and there are actually 4 FDA-approved tests, are positive above a threshold versus negative below a threshold. And in study after study after study, you find that in general, the difference is anywhere between 2 to 6-fold. Higher probability of responding to blockade of this pathway if the tumor expresses the major PD-1 partner, PD-L1. So this is already, again, proof of principle that we can guide our therapy by looking at key components of the pathway that we're actually targeting. So how do we do this to expand to the 20, 25, 30, 40 potentially interesting targets? Well, there are some great technologies. We've been very involved in developing these techniques called multispectral imaging, where you can put in different fluorescently labeled antibodies. We work with Perkin Elmer, but there are a number of companies that are now making imagers that you can label these antibody with different fluorescent dyes. And so here we've simultaneously stained, and when I say we, I'm talking about Janice Taub and her colleagues, our immunopathologist extraordinaire, for CD8, FOXP3, CD68 is a macrophage marker, SOX10 is actually the tumor marker, PD-1 and PD-L1, and you get some really, really pretty pictures. The problem is that you no longer can look with your eyes and really begin to decipher this. Imagine if we were now looking at 10 or 15 of these. These become images that your eye has a very difficult time deciphering. Now you can pull out specific sets. So for example, what Janice did here is to just look at SOX10, which is tumor versus PD-L1, and here CD68 versus PD-L1, and you can see that in this particular melanoma, virtually all of the PD-L1 is expressed by tumor cells and not by macrophages. There are other tumors that I could show you in which it's just the opposite. The macrophages express almost all the PD-L1, and it's not on the tumor cells. What does that mean? We don't yet know. There are far more questions that we don't have the answers to. But what is very cool is that the technologies that can actually give us the data, which in humans that we can now annotate with clinical courses, responses to immunotherapies that are actually focused on these targets, now begin to give us information databases that we can really learn from. This is one example of doing some simple nearest neighbor analyses that showed that, for example, in this particular melanoma, 19.3% of the CD8 T-cells were actually touching macrophages. So this is, again, sort of a very primitive analysis, and we realized that we had to do much better. This is an example for actually of a patient with Merkel cell cancer with sort of this gauntlet of PD-L1 positive tumor and macrophages that are blocking these yellow PD-1 positive T-cells from getting into the tumor. You can see there's this lone T-cell that's broken through the gauntlet. This T-cell is not going to have an easy time getting rid of this whole tumor. We now treat with anti-PD-1, and you completely blow up this PD-L1 gauntlet, and now all of these PD-1 positive CTL flow into the tumor, and the tumor regresses. But again, to take this to additional orders of dimension, we realized that we needed to become astronomers. So because the image analysis was really clearly the barrier. And so turns out that the Space Telescope Institute, which is where all of the Hubble Space Telescope images are actually analyzed, is at Johns Hopkins. So we went over to the Homewood campus to Alex Saleh, who built all the programs to analyze petabytes of pixel imagery from the Deep Sky survey. And I have to say, I'd never even heard of the term petabyte until I met Alex. We sort of showed him some of this stuff, and Janice has been really doing all this development. And literally sitting in his office, he loaded some of the data onto his computer, converted it to R, and literally in 5 minutes he did what it took Janice's group 2 weeks to do. So I think the notion that we will get to the point where we can query lots and lots of key target molecules for which there are therapies, for which there are antibodies and drugs in tumors, and develop biomarkers that will guide therapy, I think really is now no longer science fiction. So I want to finish by just showing you a vignette about another cool thing that's happening in our field, which has been the intersection, really the marriage of genetics and immunology. So again, 5, 6 years ago, if you went to the American Association for Cancer Research meetings and you went to the cancer genetics sessions, you would not find a single immunologist in that session. You went to the immunology sessions, you wouldn't find a single geneticist in that session. These days, I'm teaching all of the geneticists to be cancer immunologists. Even Bert Vogelstein, I was his first graduate student. I'm actually teaching him to be an immunologist. He's actually smarter than I realized he was when I was a student. He's pretty good at it. So what's the basis for the intersection? Well, it's actually relatively obvious. Mutations in tumors are probably the most straightforward thing that provides for them antigenicity that the immune system can use to recognize and distinguish a tumor cell from a normal cell. So the tumor's got mutations. Some of these mutations are coding. They're non-synonymous. If they appear in a peptide that gets presented on MHC, then that's a target that a tumor-specific T-cell can recognize as potentially quote-unquote foreign, at least different from a non-transformed cell. So the implication is that cancers that have many mutations therefore have many antigens and therefore will engender a stronger immune response. And in fact, if you look at this spectrum of tumors that respond better to PD-1 blockade, so we're taking those as cases where the patients have large repertoires of anti-tumor T-cells. And again, the immune toxicity rates for something like anti-PD-1 are pretty low. They're less than 5% severe adverse immune event. So clearly, when we unleash these responses, they are largely selective for the tumor. And in fact, if you look at this analysis by tumor type of mean mutational load per exome, each one of these is a separate tumor, you find that, and then what's circled are the tumors that have decent response rates to anti-PD-1. All of these PD-1 blockers are going to be either approved or going to be approved. And you can see they tend to be concentrated towards the higher end. Melanoma, very high, lung cancer, and on down. But it's certainly not perfect. One of the cancers that is considered a non-responder to anti-PD-1 is colorectal cancer. And that's the case because in Suzanne's original paper, 17 patients, her big 300-patient study, 17 colorectal cancer patients, no responses to anti-PD-1. But in the very first paper that she published, that actually people weren't paying attention to when it came out in Journal of Clinical Oncology in 2010, the first colon cancer patient ever treated had a complete response to anti-PD-1. And this really sort of irked us, and so we went upstairs to Bert Vogelstein and Luis Diaz and Ken Kinsler and our genetics colleagues. It's nice having them one floor up. And we did a brainstorming session, and they asked, well, was that responding patient an MSI, microsatellite unstable, this genetic subgroup that has a defect in a mismatch repair, in a gene encoding mismatch repair enzyme? They have a lot more mutations, so they would be more antigenic. And I can tell you, it's embarrassing to admit that this notion actually came from a geneticist. But it did. And in fact, there are a number of cancers that about 15% of colorectal cancer, smaller proportion of metastatic, about 20% of gastric cancer, endometrial cancer, even about prostate cancer, it's about 2 to 4%, pancreas cancer, about 2%, that have this mismatch repair defect. They're called microsatellite unstable. And these tumors have between 20 and 50-fold more mutations than the typical tumor, oftentimes more than 1,000 mutations. And you can see when you actually look at the spread, there are these groups that are very high. And so that led Luis Diaz and Young Lee to actually do a clinical trial of anti-PD-1 in patients with mismatch repair proficient colorectal cancer. So these guys have the typical 50, 60 mutations per exome versus mismatch repair deficient colorectal cancer between 600 and 2,000 mutations per exome, as well as sort of a basket of, not a basket of deplorables, a basket of non-colorectal cancers that were also mismatch repair deficient. And this is a waterfall plot. So basically, this is the change in the tumor mass of index lesions after the start of therapy. So if it goes up, that's bad. That means the tumors are growing. If it goes down, that's good. That means the tumors are regressing. And this was one of these cases where it only took about 24 patients to see that we had something really interesting going on here. Basically, less than a regression of greater than 30% by area is roughly 50% by volume is considered an objective response. And you can see that the majority, not all, but the majority of the mismatch repair deficient cancers respond really quite impressively, whereas the mismatch repair proficient colorectal cancers don't respond, although there is this interesting stabilizing group that, again, we think we can capture. Understanding how this works means that we have to understand the T-cell repertoire. Now, understanding the T-cell repertoire is complicated. Why? Because T-cell receptors, just like antibodies, have these diversity-generating mechanisms that you all probably learned, depending on how old you are, either last week in medical school or 20 or 25 years ago. But these diversity mechanisms give you literally, lots and lots, probably 10 to the 17th, different possibilities concentrated in this one region between the V and the C region called the CDR3 region. So now with deep sequencing, and I don't know was Harlan Robbins speaking in this? He was, yeah. So he actually has perfected the application of deep sequencing to really query the T-cell repertoire, and that really gives a whole new dimension to understanding the T-cell repertoire. The idea being that if you see an expansion of a particular CDR3 region by deep sequencing, by definition, that is a clonal expansion, and by definition, it means that the T-cell that has that T-cell receptor has responded to some antigen. It doesn't tell you what the antigen is, but it tells you that there has been an expansion. So in fact, when you combine that with functional analyses of candidate peptides that you generate based on whole exome sequencing of the tumor, you can find in, for example, the MSI colorectal cancer patients, that after PD-1 treatment, you can find lots of these mutation-associated neoantigens. We call them manna. If you're the immune system, they're manna from heaven. You can actually find CDR3 regions that you sequence out of T cells in the peripheral blood upon activation with these different peptides. You can find these clones expanded in the archival biopsy of that patient's tumor. Now, without expansion, you generally don't see them in the peripheral blood. But now in this patient, you treat with anti-PD-1, and all of a sudden, these clones that you saw in the tumor shoot up in the peripheral blood within 2 weeks, which was the first time point we looked. Now, the first regression that the radiologists saw that said, okay, you can now call this patient a responder to therapy, was not until 3.5 months later. So now, simply by looking at expansion of T-cell clones that are actually in the archival tumor, you have potentially a biomarker that tells you a lot more quickly what this patient is going to do long before the radiologist actually tells you what's going to happen. Don't worry, we're not going to put the radiologist out of business, okay? Your husband will still have a job. But this is actually very cool. And we can even now look at this in settings where we actually give the PD-1 blocker before surgery. So these are lung cancer patients that are considered operable because they don't have visible metastatic disease. But we know, based on the extent of their disease and lymph node involvement and all, that they have a 75 to 80% probability of relapsing, which means they actually do have metastatic disease. We just can't see it. The surgeon takes out the primary tumor in the lung, and there are micrometastases which come back. So the question is, can we actually expand clones in these tumors by actually taking advantage of the fact that you have your tumor in place before you do surgery, give the anti-PD-1 before surgery, induce a clonal burst in the tumor, and then maybe those clones, those T cells, do what T cells do. They're migratory. They don't just sit there. They will circulate. They'll leave the tumor, go into the circulation, and do what T cells should do, which is seek out micrometastases. And amazingly, this actually seems to happen reasonably frequently. This is actually the pathologic response, so loss of tumor at the time of surgery relative to pre-treatment biopsy. And you can see about 40% of these patients have what's called a major pathologic response, which is greater than 90% of their tumor is actually replaced with something that's not tumor. And with anti-PD-1, what it's replaced with is lymphocytes. So this tumor, you can see there are a few lymphocytes here. At surgical resection, after just 2 doses of anti-PD-1, other than this little piece of tumor here, the rest has basically been overrun with lymphocytes. And when you actually do your clonality analysis, you see that a lot of these clones that have expanded in the tumor now expand in the peripheral blood. In fact, of 100 clones that Kelly Smith found to be expanded in the blood pre versus post treatment, 94 of them were significantly increased. So 4 clones went down, 94 went up, and 60% of these, 56 of those 94, actually, in fact, could be found to have been expanded in the tumor itself. So I think we can now start to really query what we're doing, and now combining that with a functional analysis that we call manifest for mana-functional expansion of specific T cells, we can take predicted peptides, culture them with peripheral blood T cells, and now read out, instead of things like interferon production, we can actually read out clonal expansion based on CDR3 deep sequencing to actually have a more sensitive assay. And in fact, this is an example of what you see. So these are basically expansions that you see in response to predicted manapeptide number 25. This is number 1, and you can see lots of T-cell clones get expanded, and all of the other peptides in the screen are essentially the negative controls for the actual peptide of interest. And so you get a very, very specific and sensitive readout. This picks up about 90% of mana-specific T cells that you actually miss by the conventional ELISpot analysis. And so what this actually allows us to do potentially is to have a much better read on what are the relevant tumor antigenic peptides if one, for example, wanted to now develop these personalized mutation-specific vaccines, which people are now doing, where you whole exome sequence, define the antigenic peptides, synthesize the peptides, formulate a vaccine, and then treat. One of the barriers to this has always been that the prediction algorithms are highly imperfect. So now with this assay, we can apply this to actually identify potential relevant functional targets of T-cell responses to develop personalized vaccines to go along with blockade of checkpoints and various of the other immune manipulations that one might want to do. So I hope that I've communicated to you that as exciting as this transition has been for our field and what it can do for patients, that we've really just scratched the surface, and we've got a lot more that we can do that technology and our creativity will hopefully allow us to do, to be able to mine a much larger proportion. Again, finally, our program has grown quite a bit. I went back to a picture of our program about 8 years ago, and there were about 10 people. And so it's grown a bit because now in our cancer center, basically everybody wants to be an immunotherapist. So it's certainly an exciting time for our field, and really appreciate the opportunity to tell you about what's been going on here.